The first of the two specific immune processes we're going to look at is cell-mediated immunity. You'll remember that cell-mediated immunity is the means by which we manufacture T lymphocytes to go out and kill virus-infected cells and to kill cancerous cells. And I'm going to go through just a quick laundry list of the key players in the cell-mediated process. These are the most important types of cells that come into play to make this process work. We're going to see that you have to have some sort of an APC, an antigen-presenting cell. Now, antigen-presenting cells can be a B cell, but they don't have to be. Oftentimes, they are a macrophage. Sometimes they are what's called a dendritic cell, which is like a specialized type of macrophage found in the dermis, right at the borderline between the dermis and epidermis. So you don't always have to have a B cell be your antigen presenting cell, but as we will see shortly, you do have to have some cell that presents the foreign invader, the antigen, to the T cells. We also need to have some CD8 cells come into play. Those are the cytotoxic T cells. They are called CD8 cells because they have on their surface a cluster differentiation protein. That's what CD stands for. They have an identifying protein called CD8. Okay, so these are the cytotoxic T cells. The helper T cells have a different protein on their surface that marks them and identifies them called the CD4 cell. CD4 cells and CD8 cells are key players. You have to have both helper T cells and cytotoxic T cells present. We're going to see that there is a role that suppressor T cells play in this. The principal role they play is to sort of keep the cytotoxic or the cell-mediated response in check so that it doesn't go out of control and start killing off cells that it shouldn't be killing. And then there are what are called memory T cells. These are T cells that will be manufactured so that if we ever encounter this particular antigen again, that we already have a number of cells in place, ready to go, that remember how to respond to that particular pathogen. Here's a quick, very important thing to remember, and that's really, really critical. If you have free-floating antigens in the body, like let's say bacterial toxins are free-floating in the blood, or viral particles are free-floating in the extracellular fluid, T cells are blind to those. T cells cannot see any kind of free-floating antigen. The only way a T cell can recognize an antigen, the only way it can realize that an antigen that's not supposed to be there is actually present, is to have an antigen presenting cell show it to them. This is a key difference, and I'll highlight it again when we get to the B cells, because B cells they are different. They have the ability to see free-floating antigens, and T cells do not. So let's take you through the major components of a cell-mediated response. Again, the goal here is identify a cell that's become cancerous or that's been infected with a virus and find a way to kill that cell off. And there are three sort of generalized phases of a cell-mediated response. The first being the surveillance and recognition phase, followed by attack, and the last portion of the game is to generate some memory T cells for future reference. So let's go through this step by step, starting with surveillance. During surveillance, imagine, if you will, you have CD8 and CD4 cells, that's both cytotoxic T cells and helper T cells, that are found throughout various lymph nodes in your body. They're found in the blood. They're basically throughout your body, and their role is to simply check out cells. Okay, so let's take CD8 cells, for example, first. These are the cytotoxic T cells. Cytotoxic T cells, remember, each have their own uniquely shaped T cell receptor, or TCR, on their surface. That's that receptor protein that's shaped differently so that different CD8 cells can recognize differently shaped antigens. What cytotoxic T cells do is they cruise around in places like lymph nodes, for example, and as they encounter cells, they feel those cells up, for lack of a better term. Okay, they put out little pseudopods that kind of reach out and feel that cell to see, is that cell a host cell or is this some sort of foreign cell that I don't recognize? The way it knows the identity of the target cell is because of the presence of a protein on the target cell called MHC proteins. MHC stands for major histocompatibility proteins. Major histocompatibility proteins, or major histocompatibility complex, MHC. And these are also sometimes called, particularly in anthropology books, they're called human leukocyte antigen groups, or HLAs, but it's the same thing. There are two main classes of MHCs. MHC1s are found on all cells in the body, all somatic cells. Okay, it doesn't matter whether you're a bone marrow cell, you're a epithelial cell, you're a cartilage cell, it doesn't matter, you have an MHC1. That MHC1 is uniquely shaped for you. Nobody else in the world has, or very few, have a similarly shaped MHC1. That's unique to you. Just kind of think of it like your own fingerprint, your protein fingerprint on all cells. There's a second class of MHCs called the MHC2s. These are found only on antigen-presenting cells, only on APCs. Now here's an example here of a target cell. You can see, let's see here, we've got right here, I'll outline it for you. This is our MHC protein, and it says it's an MHC1. That tells us that this is just a regular old somatic cell in the body. I need to erase a few things here so we can see this interaction and what's going on. So let me get this out of the way. So here we have a CD8 cell, cytotoxic T cell, hanging out in the lymph node. Here comes a cell that it encounters, a somatic cell, a host cell. And if that cell has become infected with a virus, remember that viruses infect cells by physically entering into the cell and taking over the nucleus of the cell. And they basically force the cell to stop making all the proteins it normally makes and instead make copies of virus, and then to release those. So basically what a virus does is it takes over a host cell and forces it into becoming a virus factory. When that virus infects the target cell, one of the things the target cell does to try to warn nearby cells of the viral infection is it will take some of the pieces and parts of the viruses that it's making, and it will incorporate those into its MHC1 protein. So here is this processed antigen that was from the virus that the target cell has intentionally installed or put into its MHC1 molecule. And then it put that MHC1 out on the surface and started secreting a chemical called interleukin 1. Interleukin 1 is like the international calling cards of all cells to come on over and check me out, something's going on. Okay, so typically cells that have been infected will secrete interleukin 1 just to call white blood cells to the area to alert them to the problem. So here comes this CD8 cell. It responds to the interleukin 1. It has a T cell receptor on its surface, and that T cell receptor is going to check out the shape of the antigen that's in that MHC1. And it's probably likely that the shape of this T cell receptor is the wrong shape to bind or fit with that antigen, in which case that cytotoxic T cell will go away and another cytotoxic T cell will come in and try its luck. It will see if it has a properly shaped T cell receptor to fit with that particular shaped antigen. And this process can go on for hours until we find the right shaped cytotoxic T cell that has the right shaped T cell receptor to interact with, fit with, recognize this shaped antigen. Okay, this is all the part of the surveillance phase. When we find the right shaped CD8 cell, okay, that's an important thing because at that point we know this is the guy for the job. This is the cell that can go out throughout the rest of the body and find any other cells in the body that have been infected with this specific virus and identify them. So what we want to do at this point is make a bunch of clones of that cytotoxic T cell that's the right shape. And that's what I'm going to talk about in a second here on the next slide. So before I do that, though, one other thing I can mention here, and I have to erase a couple things so you can see it again. Remember I told you that cytotoxic T cells are called CD8 cells? Well, it was because of those proteins on the surface, that CD8 protein on the surface of that cytotoxic T cell. Here is that CD8 protein. They're representing it by this little bar-shaped thing. Once the right-shaped cytotoxic T cell binds to this antigen here, the CD8 protein kind of locks it in place, and this is what I guess kind of cinches everything together so we know, okay, we've got the right-shaped cytotoxic T cell. We should start making copies of this cytotoxic T cell. So the problem with that is if we only had those CD8 cells going out and finding, which one was the right-shaped one, if we didn't have some sort of confirmation mechanism in place, it's very possible that a cytotoxic T cell could erroneously identify some antigen as being foreign when it's really not, and start launching an attack on our own body cells. So we have a safety mechanism in place. That safety mechanism is the helper T cell. Okay, helper T cell, or the CD4 cells, if you want to call them that too. The helper T cells confirm and make sure that the cytotoxic T cell is correct in its belief that there is an infection happening. Okay, and until the helper T cell confirms that the infection is really there, that cytotoxic T cell back here is just going to be in a holding pattern. It knows it's the right one for the job, but it hasn't gotten permission yet from a helper T cell. It hasn't had the helper T cell confirm this, and so it isn't yet able to replicate. This is a little like with nuclear weapon stations, how to launch a nuclear warhead, you have to have two people each simultaneously turning a key at the same time. It's the same kind of idea. Until the cytotoxic T cell receives that confirmation or that permission from the helper T cell, it's not going to be able to clone and go out and kill cells. So the question now is, well, then how does a helper T cell go about confirming and making sure that really there is an infection present? Here's how that happens. Helper T cells are a little different in the way that they interact with other cells. Whereas we just showed you cytotoxic T cells or CD8 cells interact with host cells, those are the ones that have MHC1 proteins, CD4 cells, the helper T cells, only can interact with those cells that have an MHC2 protein. And you'll remember MHC2 proteins are found only on antigen-presenting cells, things like macrophages or dendrocytes or B cells. So we've already talked about the interaction between a CD8 cell and a host cell that's been infected, and shown that it has been infected by having that antigen presented in its MHC1 complex. Meanwhile, while that's going on, we also have helper T cells in places like lymph nodes, and they also are examining cells that are presenting to them different antigens. Now, an antigen presenting cell, this is critical, does not become infected. An antigen presenting cell intentionally goes out, finds free-floating antigens, and consumes them. And when it eats them, it's not getting infected by them, it's just eating them and digesting them into little parts. And once it's digested those into little parts, that antigen-presenting cell has an MHC2 on its surface, and it will present that antigen that it's digested in that MHC2, and then it will secrete the international calling card of all cells. It will secrete interleukin-1 saying, hey, helper T cells, come on over and check out what I just found. So when this, here it is, we're using a macrophage in this case as our example over here. When this macrophage consumes A virus. Here's the virus it consumed. It breaks it down. It digests it. It puts that viral antigen inside of this MHC2 complex. And then it secretes interleukin 1. That calls helper T cells over. Every helper T cell has a different shaped T cell receptor on its surface, just like every cytotoxic T cell had a differently shaped TCR on its surface. And we may have to go through several hours worth of having one helper T cell after another, after another, after another, come and see if theirs is the right shaped TCR to bind with and recognize this viral antigen. Eventually, we're going to find the right shaped helper T cell to recognize this antigen. When that happens, that helper T cell begins to secrete the kinds of chemicals that are necessary to confirm to the CD8 cells that an infection really is happening. So in other words, once the helper T cell has confirmed with the antigen presenting cell, oh, yep, you were right, antigen presenting cell. You found the infection, and I hear from the CD8 cells they found an infected cell. I'm going to start secreting interleukin 2. And that interleukin 2 is going to stimulate the cytotoxic T cell over here, and only the cytotoxic T cell that has the right shaped TCR. All the other cytotoxic T cells that were the wrong shape will not be stimulated. Only the one that's already bound to the infected cell gets stimulated. That cytotoxic T cell then begins to rapidly clone itself. It begins to divide very rapidly, and it makes two major groups of cells. It makes a whole bunch of copies of itself that are just cytotoxic T cells, and they are going to go out and they're going to look for any other somatic cells, body cells that have become infected, and they're going to kill them. And I will show you how they kill them in a minute. They also make a handful of what are called memory T cells. Memory T cells do not get involved in the attack. What they do instead is they go spread out through all the lymph nodes in the body, and then they just hang out. And they may hang out for decades, just surveying the passing lymph, looking to see if they ever see a cell infected with this particular virus again. Okay, so they just go out throughout tissues throughout the body and look for, in the future, any cells that might be infected with this virus so they can destroy them immediately. So you might have hundreds of thousands of memory T cells spread all over the body so that the next time you're exposed to that particular virus, you've already got a whole army of cells ready to go to clear that infection out even before you get symptoms. This first time around, though, when you get infected with a virus, for example, it takes a while before a cytotoxic T cell even discovers the infection's afoot. Then it takes a bunch of hours till we find the right shaped cytotoxic T cell for the job. Meanwhile, it takes a while for an antigen presenting cell to locate the antigen, to digest it and present it in their MHC2. Then they got to secrete interleukin 1. They got to call over the helper T cells, and we might take a few hours to find the right shaped helper T cell. When that happens, it secretes interleukin 2. That stimulates mitosis or cloning of this cytotoxic T cell. And it takes a number of hours to clone enough copies of that cytotoxic T cell. So the first time you're exposed to an infection, it's going to take anywhere from 3 to 5 days before you really can launch an attack against that virus. Meanwhile, the virus has had ample opportunity to replicate inside your body, and as a result, you're probably going to have all kinds of nasty symptoms related to that infection. Okay, during the attack phase, this is after we've made all our clones. We've got all our CD8 cell clones. They spread out throughout the body, and what they do is they go around feeling up host cells, remember somatic cells, the ones that have MHC1 on their surface. And if they find a cell with MHC1 on its surface that has that right-shaped antigen presented in its MHC1 complex, that copy of the cytotoxic T cell will perform something called a touch kill. And what a touch kill is that this cell will literally reach out with a little extension of itself, a little pseudopod extension, and it will deliver a host of chemicals to the surface of that infected cell. And as a result, a bunch of holes start getting punched in this infected cell, and the cytoplasm leaks out of the cell, as you see here, and that causes the cell to die. The type of chemical that gets delivered to these cells is called perforin. It's called perforin because it's a protein that literally perforates or puts holes into the cytoplasm of the cell. So here's a real picture of a T cell, a CD8 cell here. Here's an infected cell in your body. In this case, this is a cancerous cell, but it's the same principle. As soon as that CD8 cell comes in contact with that infected or cancerous cell, it then performs that touch kill, and you can see that we get the destruction of this cancerous cell. This is a process that we haven't learned about that happens at the same time as the touch kill. It's a process called apoptosis. In upper division classes, you'll learn more about that. Now, remember, it's only the right-shaped helper cytotoxic T cells that perform this touch kill. And I want to mention one other thing, which is not only do we make lots of clones of the CD8 cells, the CD4 cells, the helper T cells, also make lots of clones of themselves. And that's so that we can release even more and more and more and more interleukin-2 so that we can stimulate greater and more rapid cloning of the CD8 cells. Again, I said that there were also memory cells that were manufactured during the time of cloning, and those memory cells include both memory helper T cells, memory CD4 cells, and memory cytotoxic T cells. And again, those then are spread throughout the body. Here's that memory T cell, here's that, and they didn't show the memory helper T cell, but there are also those as well. Those get sent out all over the body, and they remain in place for years. And their job is simply to keep an eye out for this infection should we ever come in contact with it again.